Hello everyone, welcome back. In this video, we are going to start delving into inferential statistics, and this video is really going to help you be prepared for our future videos for some basic concepts that we need to talk about. So first, let's define inferential statistics. And inferential statistics give us a probability that a conclusion is true within a population that we are studying. This is different than descriptive statistics, which basically just describe the data. We get what's called the p-value, or the probability, from statistical tests. The basic process in inferential statistics starts out with defining the null hypothesis. The next step is we run a statistical test. This is going to give us some test statistic, which will eventually convert to a p-value, and then you can reject the null hypothesis if the p-value is less than the alpha. Basically, everything that we're going to be doing over the next two class periods comes down to this. We state the null hypothesis, we run the statistical test, and we reject the null hypothesis if the p-value is less than the alpha. Now, of note, the p-value doesn't necessarily tell you the effect of the intervention. For example, you can have a statistically significant p-value for one drug that reduces mortality by 50% and another drug that reduces mortality by 20%. However, it's only going to tell you that the result of the study was not due to chance. Which brings us to our first objective. Our first objective is really an overall objective for what I want you to be able to complete after these two class periods, so don't be worried if you don't have that quite yet, but you should be able to choose the appropriate statistical test given the type of data of the dependent variable. We'll talk about what the dependent variable is here in a second. Number of groups analyzed, whether data is paired or unpaired, additional factor analysis, presence of confounding factors, distribution of data, and number of patients. So this is a lot of different things to think about, as you can see. So we're going to go through these step by step, and I made an easy figure for you guys to follow. This is really summarizing a step-by-step process of how you can choose the correct statistical test. So let me pull that up. So this is a decision tree for helping you choose the correct statistical test. We'll be going over all of these statistical tests at the ends of the trees and how to get there. So as you can see, the first thing that you'll do is you'll start out with what type of data is this? Is it continuous data? Is it ordinal or skewed continuous data? Or is it nominal data? The next thing that you'll be asking is how many groups were studied? Were there two or more than two groups? Was the test paired or unpaired? And we can see paired or unpaired, paired or unpaired. And then some additional points can be taken in for certain statistical tests, like the number of factors that are looked at, or the number of patients within certain cells. I would highly recommend keeping this chart with you pretty much all throughout pharmacy school. This will really help you identify if they're using the correct statistical test in a lot of different studies. It certainly doesn't cover every statistical test that has ever been developed, but it's pretty comprehensive, and you're probably going to find over 80% of statistical tests that are done within this chart. So the first step in using this figure is to decide on what type of data the dependent variable is. So let's quickly define dependent versus independent variable. So our second objective is identify the independent and dependent variable within a journal article. The independent variable is going to be the variable that affects the outcome. So this variable is typically the drug. This is usually in a randomized control trial. This would be manipulated by the investigator. So this is the drug, and this is going to affect the dependent variable, or the outcome of the study. So for example, if we were looking at the effect on a medication on weight loss, the medication would be the independent variable, and the dependent variable would be weight loss. So our next objective to work through that figure that I showed you is to be able to identify the scale of measurement of the dependent variable. So identify the scale of measurement of the dependent variable, and this is going to be classified in three major categories, and the first one being nominal. Nominal data is separated in the categories, and there's no rank or order to these categories. So this might be like red, blue, green. Those are all different categories. There's no saying that red is better than blue, etc. If we're looking at going back to our weight loss example, we could use a percent of patients that achieved 20% weight loss. They either achieved 20% weight loss or they don't. Those are the only two categories. So that would be a type of nominal data. Ordinal data uses order, not surprisingly by the name, but difference between ranks is not equal. In the medical world, one commonly cited example is the pain scale. For example, a pain scale of 4 does not mean the patient is experiencing exactly half the pain of the patient that has a pain scale of 8. Now, however, if there are a large number of ordinal groups, sometimes statistical tests that are used for continuous data can also be used for ordinal data. However, that's more of an exception to the rule than something that occurs frequently. So if there are a large number of groups, i.e. greater than 7, may be able to use continuous statistical test. Now let's go to our last type of data, and that's continuous. And this can be either interval, which means 0 does not mean absence of whatever they're measuring. For example, 0 degrees Celsius is not the absence of temperature. And ratio is that 0 means absence. And the way that I remember this is that there is a O in the word ratio, we see right here, so that means 0 means absence. That's just how I remember it. However, when we are choosing the statistical test, usually we don't have to differentiate between the two. Usually we need to just know if it's continuous data or not. So either one of those would be okay. And our third objective will be identify if a data set is normally distributed. Normal distribution also has another name. It's called the Gaussian distribution. This is used very frequently in statistics. It occurs commonly throughout the world. So let's talk about characteristics of normally distributed data. And one is that the typical bell-shaped curve. And also the median, mean, and mode are all the same. The curve itself can be folded on itself because it's symmetric. So the third is the distribution is symmetric around the mean. And the fourth is that the skewness and kurtosis are 0. Skewness is when the data is skewed either to the left or the right. And kurtosis refers to the peaked nature of a distribution. And I'll show you some pictures here. So skewness and kurtosis are 0 when described. And then the total area under the curve, or AUC, of the total distribution is equal to 100%, or 1. So let me show you some pictures of these characteristics. So on the top here, we see the normal distribution, we see the bell-shaped curve. As we go down, we see two different skewed distributions. The one on the left here being positively skewed because you see the tail goes in the positive direction on the x-axis, where the negatively skewed data goes in the negative direction on the x-axis. And below we see positive kurtosis means that it is more peaked, and negative kurtosis means that it is flattened. The problem is that we don't always have the information that we need within a journal article to determine if the data was normally distributed. Those typically just tell us the statistical test. Fortunately, there are a few things that we can do to help us identify if the data was normally distributed or not. The first thing that we can do is we can look at the mean, find mean and standard deviation of the primary outcome. And we can use this to calculate something called the coefficient of variation. This is simply the standard deviation divided by the mean times 100%. As a general rule, the data is not going to be normally distributed if the standard deviation is bigger than the mean. The coefficient of variation is useful for presenting and summarizing information, but it only works with positive data. The most important thing here is to make sure that the standard deviation is less than the mean if they're using a statistical test for data that needs to be normally distributed. The second thing that you can do is apply the central limit theorem. The central limit theorem states when equally sized samples that are sufficiently large are drawn from a non-normal distribution, the mean of each sample will approximate a normal distribution. However, if outliers are the cause, this will still not be normally distributed, so an outlier could not have been the cause of the non-normal distribution in the first place. What does sufficiently large mean? It means a sample size of greater than 30 or degree of freedom greater than 20. And we'll talk more about what degrees of freedom are in a future video. To show you really a visual of what the central limit theorem is, I'll run through a few examples on a website here to sort of show you what this means. So I'm going to click on the link here. This link will take you to a Java application where you can test the central limit theorem yourself. So remember, it said that we could take a set of non-normally distributed data, and if we took the means of that data, it would become a normal distribution as long as we had an adequate sample size. So let's make something that is relatively non-normally distributed. So we can do this here, and we'll make sure that this has a large tail to the right. So the mean and median should not be the same. So we can see that the mean is in fact greater than the median, showing that this is probably a right-skewed distribution. And we see that the data does show some skewness, and it has some negative kurtosis there. Now, what we'll do is we'll do this in a few different ways. So there we can choose different samples. Now notice I only clicked on a sample of 2. So if we run a mean on this entire sample 5 times, it's going to show us the distribution down here. Now, doing something 5 times isn't incredibly useful, so if I click on 1,000, it will give me a distribution. Now, however, this is not a normal distribution right now. Notice that the median and mean are still different. There's still some skewedness there. And it's probably because I chose an n of equal to 2. So let's go with an n of equal to 10. Remember, we talked about a sample size of 30. So we'll just do 1,000 again. And now we see that, well, it's starting to get closer with an n of 10. So the median are getting closer. It looks more like the bell-shaped curve. Let's increase that again. Let's go up to 25. And we'll do 1,000. And we'll do 1,000 more. And the more tests that you do, the more it's going to approximate a normal distribution. So as you see, as we get closer, as we do more and more tests, it's going to look more and more like a normal distribution. And this is why the n really needs to be greater than 30. You can also click Fit Normal, and it will show you the normal distribution curve next to your distribution itself. So this is why when we do a test in a population that may not be completely normally distributed, we can still use some of the parametric test as long as there's adequate sample or adequate degrees of freedom and the standard deviation is not greater than the mean. If any of those violations occur with continuous data, then it's going to be skewed data and we will not be able to use a parametric test on that data. We'll probably need to use a ordinal test to analyze the data. Our last objective, and we'll call this objective number 4, is identify whether a paired or unpaired statistical test should be used. And when we talk about statistical tests, there's 3 main types that I consider. There is the one sample test. This is a test that compares a sample that you have to population-derived data. Now, this isn't going to be commonly used within comparative research, so we won't really spend a lot of time talking about these tests. We'll spend most of our time talking about statistical tests that are used for comparative testing, and that's going to be either unpaired or paired statistical test. So we have unpaired, and unpaired is when the data that is collected is independent of the other groups. So if we're looking at 2 groups and we have one group receiving a drug and the other placebo, there can't be any overlap between those 2 groups. That's in contrast to the paired statistical test, and that is within groups, or paired, and actually a way of saying unpaired is between groups. And for paired statistical tests, the data is dependent. So for example, we could do a pre and post test. We take a group of patients that have asthma and we evaluate where their knowledge is at prior to educating them. We educate them, and then afterwards we see how much knowledge they've gained. In both groups, the same patient is being studied, so you see that the group data is dependent on one another. Another example, so the first example is the pre-post test. And the second example is the crossover study. In the crossover study, a patient is on a medication, they will go through some type of washout phase, and then they'll be put on either the other medication or a placebo. Vice versa, patients that were on a placebo will be put on the active medication. This is also known as repeated measures. Repeated measures increases statistical power and reduces error. This makes sense because you're using the same patient in both groups. So the only thing that's really changing is whatever intervention you're doing. So it makes the data a lot cleaner and there's less things that could distract you from the real truth. Our next video will go over how to calculate degrees of freedom.